The final piece of the puzzle for Unit 4, Topic 1 is knowing everything we know about how DNA works. How can we use this to our advantage? And the study of that is all about biotechnology. So, biotechnology specifically looking at technology that makes use of, well, biology. So, it's pretty straightforward. We're going to specifically look at basically DNA and gene technology because obviously that's what this topic has all been about. Now, some of the things that is in this topic are quite complicated, but keep coming back to the objectives as we always do so that you know exactly what you need to know. And you can see that we've got describe, recognize, explain. So it's not really over the top. What we're going to focus on at the moment is, first and foremost, what is biotech, just to set the scene, and then we're going to look at recombinant DNA or bacterial DNA transformations. That's what we're going to start off with. Okay, so what is biotech? As I said before, it's the use of an organism or part of an organism to make a product or a process that we can use in a technological way. We use it every single day. Okay, making alcohol, for example, using yeast to ferment grains or fruits to produce alcohol is a biotech application. Production of penicillin is a biotech application. There are a lot of different biotechnology applications, and they are often used in probably less well-known fields, such as in cleaning up environmental pollution. There was talk of after the Deepwater Horizon oil spill in the Gulf of Mexico that they were going to use certain bacteria and or fungi to be able to break down oil should a spill like that happen again. But biotech is just using an organism or part of an organism, aka its genes, to for a technological purpose. In terms of examples, I'm going to sort of whiz through the main examples that we're going to look at in terms of our DNA technology, because most modern biotech, the cutting edge, I suppose, of biotech is DNA technology. And DNA technology is specifically using genes and DNA as a form of biotech. We can use those in GMOs, we can use them in gene therapy, we can use them in genetic diagnosis, we can use it in production of drugs and other things as well. So it's incredibly useful playing with this DNA and genes. DNA cloning or recombinant DNA technology, DNA transformation, gene transformations, all very similar. It's when we can make clones of a DNA fragment, a particular gene that we might want, and then we can insert that DNA into the DNA of a bacteria, a plasmid DNA of bacteria, which you should hopefully remember. Plasmid DNA is small circular bits of DNA found in prokaryotes that are basically like bonus DNA. They give them, extra powers over the basic stuff. So we take this plasmid and we put some DNA into it, and then we can basically con that bacteria into making us whatever we want it to make. And this is actually used in order to make insulin for diabetics, so they can cause bacteria to start producing insulin that they wouldn't normally produce. As a result, we can make plenty of insulin to then sell on the market. Another type is PCR, polymerase chain reactions. So basically, this is just DNA cloning. It's taking a tiny bit of DNA and it's making lots and lots of copies of it. Okay, it's taking the DNA, it's sticking it in the photocopier and hitting, 300 million copies. By doing this, by taking these copies of the DNA from an original bit of DNA, a small bit of DNA, we're able to more readily analyze that DNA. Whereas previously, if you've only got a little bit, well, tough luck, because there's not much you can do with a little bit of DNA. So only from trace amounts of DNA, whether it be from at a crime scene, or whether it be that we're trying to use a certain gene in the lab and we need more of it, PCR can then replicate that DNA, over 1 million times and create lots and lots of copies. Gel electrophoresis often goes hand in hand with these other techniques because it's a visual analysis technique. It allows us to see basically what DNA we're working with. So researchers can take some DNA that they've put through PCR to try to PCR, my apologies, to try to multiply it, and they can take that and they can have a look. Well, what DNA has it actually produced by splitting the DNA up into fragments and then basically using chromatography and a little bit of electric current to split the DNA up, and then we can analyze it. Other techniques which are sort of more analytical, I suppose, DNA sequencing allows us to be able to directly sequence some DNA. This is used in small scale if we have a small DNA sample, or large scale. The Human Genome Project took the human genome, all of our DNA inside our cells, and completely sequenced it. So they had a complete map, a fully written down copy of human DNA. So it allows you to see exactly what bases in what order is in that DNA. And then that can also go alongside things like DNA profiling, which is sort of an abbreviated version of DNA sequencing. So that's setting the scene for you there. That's what biotechnology is. We want to look at one particular one in this video, and that's making recombinant DNA or DNA cloning. So DNA cloning is a molecular biotech technique that makes many identical copies of a piece of DNA, which, easy example is a gene. It takes that DNA and cuts it from wherever we originally found it and inserts it into the plasmid DNA of a bacteria, making recombinant DNA. Recombinant coming from recombine. Okay, we're cutting open this DNA, we're sticking in a new bit, and then we're putting it all back together again. We then take that recombinant DNA in the plasmid and we put it back inside the bacteria that it may have been taken from, or we put it into some new bacteria. Then that bacteria has the genes that we want. It can then make the protein that gene codes for. And if that protein is something that's useful for us, then we can reap the rewards. We're basically conning these bacteria into becoming our own little factories. And those bacteria will then continue to reproduce in large numbers and quickly, as we know bacteria do, making more and more of these little factories. Okay, so recombinant DNA technology is transferring a gene from a member of one species and then inserting it into the genetic code of another species. Plasmid DNA doesn't replicate with the normal chromosomal DNA the same way that we might expect it to. And a single bacteria cell, let's say just an E. coli, for example, can make heaps of those little plasmids. Those plasmids, the more there are, the more they're expressed. Not only that, it can also pass those plasmids on to other bacteria that don't have that plasmid. So we could take just one transformed bacterial cell and even without transforming them large scale, potentially produce a lot of bacteria with many copies of this plasmid. The more plasmids there are, the more we end up getting our end product. We can split this process basically into 4 steps, okay? Number 1 is taking the DNA, okay, from wherever we get it from, okay? So the target DNA is cut out from, or the target gene, say, is cut out from its source DNA, and we open up a plasmid wherever we've gotten the plasmid from. The target gene then gets inserted in, okay? So we've cut open the plasmid, we've put in this gene that we want into the plasmid, okay? And then DNAligase, which we should remember from DNA replication, seals it all up. We now have a plasmid that has all of its original genes plus our new gene that we've inserted into it. The transformation process then adds that recombinant plasmid into the bacterial cell. So we coax the bacterial cell into soaking up this recombinant plasmid, and then the bacteria, being thoroughly and sufficiently tricked, goes on and makes many, many more copies of that recombinant DNA, many more copies of the plasmid, and then passing it on to its friends. Looking at these processes in a little bit more depth. So step one, we require something called restriction enzymes. Restriction enzymes are specific enzymes which are able to cut DNA at certain sites. We get these from bacteria. Bacteria naturally produce them because they have their own uses for them. And the good thing about these restriction enzymes, what makes them, I suppose, the most useful, is that they cut the DNA only at specific restriction sites. So they look for a particular code of base pairs, 4 to 8 long, and that is the, I suppose, the dotted line for them. That is where they know to cut. So once it cuts that, then we end up with our section of DNA we know as a restriction fragment. You can see in the image there that it's showing that it cuts it, at a particular sequence. The benefit of cutting it at this particular sequence is that when we want to stick it back together, we're going to have complementary base pairing. Okay, if we cut both our target gene out using the same restriction enzymes as we do to cut the plasmid open, then they're naturally going to stick together. We have two different ways we can use a restriction enzyme, or two different ways they cut. If they cut, leaving an overhang, as you can see in the image, that's known as a sticky end. Or if there's no overhang, that's known as a blunt end. Different ones will be used for different purposes. Neither one is necessarily better than the other. So we use restriction enzymes, we cut out our target gene from wherever we've gotten it from, and we cut open the plasmid, okay? As long as we've used these same restriction enzymes, then that plasmid and the restriction fragment are going to fit beautifully together. So DNAligase is therefore able to come along, fit them together, glue it all up, and it's like it never happened, okay? Sealing up those sticky ends or blunt ends, as the case may be. So the result of that after this second step is that we have our recombinant plasmid. Step one, we use restriction enzymes to cut out the target gene, e.g. insulin. We use that same restriction enzyme to cut open the plasmid, and then DNAligase takes that target gene, the restriction fragment, and glues it in with the recombinant with the plasmid to make the recombinant plasmid, which is now just one gene bigger than it was before. The next step is the transformation. We have our gene now. We just need somebody to pick it up. And that's step 3 here is known as transformation, because it's the step that takes this bacteria from not having it to having it, okay? Generally, this is done by giving the bacteria a shock, usually a heat shock, because when we give them a shock, that causes them to take up the plasmid. They absorb it into their cell, into their cytoplasm. So what we typically do as well with this is we not just give them the actual gene we want them to have, we can also put in an antibiotic resistant gene, which helps us later down the line, because then what happens is when we've seen, if you're following through this image here, when we add the DNA that we've created into the bacteria, we give it a heat shock, right? Now we've got a whole bunch of bacteria that have hopefully taken up that plasmid, but we only want the bacteria that have taken up that recombinant plasmid, because we're going to then go off and grow them and culture them and get lots and lots. There's no point putting time and energy into raising bacteria that haven't taken up the plasmid, because not all of them, after undergoing heat shock, will actually take it up. So when they insert the target gene into the plasmid, they often put an antibiotic resistant gene in. So what that means is after we've gone through this whole transformation process, we can then treat our hopefully transformed bacteria with antibiotics. Now, those that have been successfully transformed will survive, and those who haven't been will die. Okay? So then we've now got a whole bunch of transformed bacteria here, which are going to act as little factories for our recombinant plasmid DNA and also the protein or product that we want to make. Step 4, profit. We're going to take that, okay, we're going to grow these bacteria, these transformed bacteria, into a large culture and get them to express that gene. As they do, if you're using the insulin example again, okay, as that mRNA gets transcribed, translated into the protein, then we take those bacteria, we kill them by bursting open the cells, and then we can filter it through different types of filtration methods in order to get the protein out. Okay, the bacteria have served their purpose. We kill them. We take the insulin for ourselves. And that is our recombinant DNA transformation process. We will actually get to experience this in the lab. Okay, but if nothing else, just remember this slide, these basic 4 steps. Cutting the DNA open, inserting the DNA, the target gene, and then sealing it up. Shocking the bacteria into absorbing that transformed DNA, and then using the bacteria to make what we want. And that is DNA transformation.